Good afternoon, and welcome to another Ageless webinar. Today, I am here with Johnny Adams and Kyle Brewer. Johnny Adams is active in the Age Reversal Network and the Perpetual Project, a new volunteer association aimed at reversing degenerative aging, one of the most problematic things on the planet today. He also leads the Aging Intervention Foundation and the Gerontology Research Group, where they focus on a wide variety of collaborative things, all centered around a common theme that's really directly aligned with our mission, which is to, and when I say our, I mean Ageless Partners, to reverse the aging process as soon as possible across all 78 organs in the body and the microbiome. We also have here Kyle Brewer, who is the founder and CEO of ETA Biotechnology. They are also targeting aging across multiple tissues using a very interesting approach, with an emphasis on small molecule drugs. And so today, Johnny is going to be presenting on a very interesting topic. This is this is the second of a series of gene therapy webinars. And today we're going to go through genomic analysis. We're going to look into a wide variety of different concepts and tools. And it's going to be very interesting because what we want to know is now that these tools are available, now that we can actually sequence our DNA, the question we have to ask ourselves is, now what can we do with that information? How do we benefit ourselves and our families to use these tools to live healthier and to live longer? And so thank you, Johnny. Really appreciate you coming on today. And yeah, feel free to share your screen and take it away.
All right. Thank you, Jason. You will find the information I'm going to share with you today very useful. And although I've been working for many years to solve aging and keep us young and help with projects to analyze and annotate gene papers and similar initiatives, only recently have I taken a really deep dive into the main topics that I'm going to talk about today, like genomics, gene analysis, and to a lesser extent, gene therapies, and even a little on genetic counseling. It's a massive field, and you can learn for years and still just scratch the surface. In addition to genes known to improve healthy longevity, one of my many goals is personalized gene therapy. And that would start with an individualized genomic analysis with some of the kinds of tools you'll see today. And then a custom set of gene therapies would be developed, suited or designed to the individual and their genetic improvements. So that's just one idea, though. So before we get into the main event, I'm going to share with you my background. I'm going to rip through this pretty fast. This is from my regular presentation that I give. And I am hoping to, in you, the viewer, to spark motivation and perhaps even ignite the imagination you had when first starting. Our work is very hard, and we have some very, very great things ahead. So I'll now share my screen. First caveat, this is not medical advice. And today we will cover a bit about me and what I call the star shot. I'm available to help scientists and companies solve aging and keep us young. At the top of the list, gene therapy, and also at a close second, resetting the epigenome to a more youthful state, which some believe are synonymous to cellular reprogramming, and some others. And we'll get into some reasons and motivation why we do this. And then we'll get into some longevity genes. And I want to say regarding gene therapy, I often hear there are so many problems with gene therapy, and I just want to say we're in the early development stages. We are about solutions. Let's solve them. Then we'll take a look at the OAK-VAR system and their built-in report, which covers things like longevity, polygenic risk scores, longevity variants, longevity drugs, and major risks. We'll take a look at different resources and courses you can take to learn more. 2 top organizations where you can get your genome sequenced, some genome browsers and some other tools, and then Q&A and open discussion. So first, a little about me. This is my main Aging Intervention Foundation website. You'll see these links again a little later. Good information here. This has been years in the development. This is another site devoted to mostly aging metrics, and it's my invitation to other organizations for me to help them. I'm very selective in the scientists and companies I work with, and only interested in pouring my time into those active in solving the problem of aging so we can stay young. And there are some interesting things here you might want to explore some of these. One of these is something that we talked about last time, protein clock. This is one of the links off my site, Aging Intervention Program, System, Therapies, Metrics. The most important part are yellow highlighted. This is in my own age management program, recent therapies and highlights of what I've done. There are a few highlights here, but at the top, most recently notated is my experience with Jardiance, the SGLT2 inhibitor, and some numbers, how it reduced glucose. I had a Clotho test, and the test results were pretty darn good. I think I'm living right. Also, therapeutic plasma exchange, and this is my results and experience. Of all the therapies I've done, this is the one that had the best, both objective as well as subjective results. Multi-talented high-level assistant for your organization active in solving biological aging to keep us young. I'm going to rip through this PowerPoint as quickly as possible. These are the websites you just saw. I'm active in the Age Reversal Network and the Perpetual Project, a new volunteer association dedicated to reversing degenerative aging. That's supported, both of those are supported by the Life Extension Buyers Club. And also I help many other scientists and initiatives. And I lead the Aging Intervention Foundation and the Gerontology Research Group. Mission: increase health span and lifespan and make the world a better place. I call it a star shot for open-ended life. And of course, healthy, vigorous, filled with joy and service to the world. I hear about moonshots. Well, been there, done that. A star shot, many, many moonshots. It's the biggest challenge, the biggest adventure, the biggest dream any human has ever had, in my estimation anyway. Some things we don't want. We don't want this. We certainly don't want this. Straight talk about what it's like to get old. You can stop this tape and read these if you want, but kind of looks like this and it reads like this. This nice young lady on the right, she's, still smiling, fantastic. But we would like to help these poor, frail people who are dependent on others. We'd like to help them get younger and lead much better, more just happier lives. More straight talk. Notice it ends and then the big sleep. Okay? If you love your money, well, park it at the door at the end. End results we seek. This is very important. We need to keep our eyes focused on the end results we want. You know, it's wonderful to have all this various kinds of testing and these programs and meetings with people, you know, free exchange of information. That's great, it's fun, but all that needs to be for a reason. And yours may be different from this, but to my mind, it's defined as physical. And you can stop the tape and read these if you want. More physical, these are practical examples. Cognitive and mental function, and then emotional and what you might call spiritual. How are we going to do that? Okay, the most important, the top of my list, gene therapy. And I have great interest and faith in the potential of knock-ins and specifically plasmid mini-circle gene therapies, and also analyzing an individual genome and personalized gene therapy. Also, as I mentioned before, resetting the epigenome or cellular reprogramming. Next, here's some other ways to do it. I'm going to bypass and not read them, but you can stop the tape and read them. Okay, it's kind of like this. I'm not quite this far out on the curve yet, but I'm out there pushing. We all are. For the eventual end result of something like this. Big question, why? What will you do with all that extra time? Well, I'm going to assume we're going to get pretty bored standing around on one leg on some beach somewhere, although we might want to take a little bit of a vacation before we get back into solving some of the big problems of the world. Find a purpose, be a force for good. All right, what's it going to take to do it? Attention, scientists who are aging solutions creators, I can help you. And others active in aging, innovators, investors, visionaries, and others, let's partner up. Here's how you can reach me. Mark your calendar. You are invited to my big 100th birthday party, December 17th, 2049. I want you there. Time marches on. So now I'm going to get out of this into the main event, which is the thing you presumably came for, and move on to the genomics and gene portion of it. There are longevity genes that we might want to edit. Here is a link, and I'll take you to each one of these, but these are different individuals' lists that I've collected over the years. We'll take a look at George Church's, Olga Borisinova and Anton Kulaga, and others in their group, in the Oak Bar, another group, are doing some outstanding things. We'll take a look at that. One of our GRG members, Brian Bishop, had created a list, and also here's a genome-wide association study that has a list. So let's take a look at those. Okay, this is George Church's list. It's online with the link that I showed you. And a brief description in the third column of what they do. Oh, low aging. Hey, I'm all in. Bone loss resistance. They go on and on. You can read these for yourself. Here is the other list by Olga and Anton, and probably a lot of other top scientists help put this list together. I think this is Brian's. Gene candidates, here it gets on myostatin inhibition, CD47 suppression. It goes on and on. Allergies, well, we tend to develop allergies as we grow older, so we might want to do something about that. Sense of smell, okay. Pain sensitivity, those things, we can have problems with those as we age. Protective mutations for heart attacks, yeah. Malaria resistance, well, yeah. You know, I might want to go down to the tropics. Taste, sprinting, and endurance. Hair color, well, Height, yeah, I'd like to be taller. What the heck? Hearing, okay. Cognitive, now deal me in, okay. And many others in this outstanding work. Here is the study I told you about, a genome-wide association study, etc. You can read it. It's outstanding and rather groundbreaking. And yeah, well, that was the end of that. Okay, so that's going to be the next section, which is the OAK-VAR. Okay, let's take a look at the OAK-VAR system. This is the link to it. I've already queued it up. And this is the main page. Here's some good information about it. You can study that. To analyze one of the genome files that you would get when you get your genome sequenced, you would click on the submit link. You'd click here and you'd browse to the VCF file. Usually there's a VCF extension, although there are others. I've only done these for several associates with VCF file extensions. You click in there and you can scroll down and choose which analysis modules or which annotators you might want to add to this. And they appear here. But let's clear this. What's beautiful about this system is that we can go up and click the longevity 2 selection, and it automatically puts those into the selections for you. There is some good documentation getting started, for example, and it shows you the steps that I just showed you. Also, viewing reports, and this tells you a little bit about the report that we're going to see next. In their report, it covers the longevity, polygenic risk scores, longevity variants, longevity drugs, and some major risks. One of the individuals whose file I ran through this system gave me his permission to show his information or his report anonymously. So let's take a look at that. This is what the built-in report looks like. Talks about it here. And it talks a little bit about how the longevity risk score was derived and how to read the result, and a little background information which you can read about from, well, let's redirect from this source, okay? But we'll go back. Longevity risk scores. And this is the link you can go to find out about it. How to read the result. Talks about it here, and then it talks about some components of it, and these are a little bit different for each subject. Okay, the next section is longevity variants. So these are variants specific to this subject. And you can click on either any of these RS numbers to go to get more information. And if you hold long enough, it takes you to this National Library of Medicine resource to gather more information. Okay, next. There's quite a lot of lists. Long list. It's weighted depending on its importance. And here are longevity drugs, and specifically, here's the description. You can all read about this yourself as to what this report does. And here's the report. Major risks. This is this subject's cancer report, which would indicate upon drilling deeper with expert advice, perhaps a genetic counselor or similar expert. These genes for this subject would relate to cancer. These would relate to coronary artery disease, cardiohereditary, and that's the end of this subject's report. Here are some top organizations where you can get your genome sequenced. This is Nebula Genomics. A number of weeks ago, I did my sample and set it in, and I would have loved to show my own report today, but I haven't received my report yet. They're still in process. And you will see the link in the document I showed you. Also, another one that I haven't done is Dante Genomics, and this is their website. Here are some genome browsers and tools. Anuj, my genomics coach, uses UCSC, and he speaks highly of it. And I'm impressed with it, of course, as well. And this is one page from it. This is a genome browser. There are many genome browsers out there, but this one gives a lot of nice information and allows you to customize the content you're seeing. Here's another resource. Well, this is Longevity Map. It's human longevity genetic variants. This is another genome browser, Ensemble. It's pretty well known. This is one I believe we looked at before, the National Library of Medicine's DB-SNP. And I've been immersing in all this, but I cannot claim yet to be an expert. But I get, if there's a particular problem or a question, I know where to get the answers quickly. And like many of you, always learning. This is SNPedia, which is a very recognized source for SNP information. Okay, next, you might want to have a chat with a genomics counselor. This is probably one of many resources I found. I'm not endorsing this, but it's something that I'm going to do once I get my genome analysis results in. This should be the page for it. I had it queued up. Yeah, DNA visit, that's what the page looks like. There are different resources and courses you can take to learn more. One is Interpreting Genomic Variation Fundamental Principles. And here's the link to it. This one I'm taking right now. I'm in the middle of it, learning a lot. This is the topics it covers. Another one, genetics counseling course through UC San Diego. I just signed up for this today. This is the page, and let's see if yeah, that's my order confirmation, so I'm in. It would be nice if certification were through a really, I mean, UCSD is major, but, you know, also John Hopkins, they have a reputation, and they have what looks like great ones. There's a 9-month course, cost is $25K, and this is the link to it. Their page is here, and they have two that are of interest. The one that was of interest to me was the Postmaster's Certificate in Sequence Analysis and Genomics. To do this justice and to take advantage of meeting the people there and networking with the intention of partnering with them to create solutions, you'd have to go there. You'd have to live there for the full course. But also, in addition to lots of other great courses, they have a Master of Science in Individualized Genomics and Health. And I've been looking into Stanford courses and others. So what questions might you have? Let's open it up to Q&A and open discussion, and I will stop sharing my screen.
Absolutely. That was excellent resources there, Johnny. Really appreciate it. That was phenomenal. There, it really shows that there's just so much resources out there. And I mean, even, what you showed, I'm sure, is a small snippet of all these organizations and universities that are offering courses, and so much resources out there for people to learn and for people to get their hands on this stuff. And you can now get your genome sequenced for fairly low cost through those different companies that you showed, like Nebula Genomics, for example. And it's phenomenal. And so one question I've been thinking about is, so, we have this gene map, let's say, and we're now developing the tools to target specific genes within the genome, which is in every cell in the body. So when we put something in, let's just say we do a gene therapy, it goes into the blood, we alter, some cells, and then within the blood, then I guess at that point it would be integrated into various muscles and tissues that as the blood is kind of circulating. But what I kind of wonder, I mean, say those cells now have different DNA, it seems like it would be hard to measure. Like we wouldn't know which parts of the body necessarily now have that new DNA unless we could really isolate in the proteome or the transcriptome in the blood to determine there's been an actual change in functionality. Am I understanding that correctly? How are you guys kind of thinking about it?
Well, good question. And I believe Kyle may have some thoughts on that. But if I may take the floor first, yes, definitely. One such tool is something that we talked about last time, and I briefly touched on the proteome analysis, or a protein clock. But I'm, more than anything, and this deserves a whole section, and I'd be happy to come back and talk about this time. All these blood tests and analysis are wonderful and an important part of our work, but in recent years, more than anything, I've been focusing on functional tests, measurements of the end results, or results that are much closer to the end result we seek. For example, something I rely on is an exercise stamina test. And I can't really go into all that right now, but also others that are physical tests that are tried and true, like grip strength, the 6-minute walk test, 30-second chair stand, things like that. Also cognitive, one of the end results we seek is our cognitive functions. And let's face it, this whole machine that we occupy, although closely interrelated with the brain, really it's the brain and our cognitive functions and sense of well-being and mental functions and so forth that we are trying to preserve for a long time.
Yeah, so I agree.
Else?
Yeah, I agree completely with you. I think these measurements that really look at outcome are better. So we have really good tools that are scientific now that can maybe predict age, such as the DNA methylation clock from Steve Horvath's lab, that is kind of the state of the art and what people look at most. But kind of the opposite of that is what you talked about, the 6-minute walk test. And there are some people working on outcomes that are done by wearables, for example. So I think this would be something. For example, there was a paper from George's Jansen's group that showed that you can have a wearable that can predict someone's age within a few years. So this is something that's completely non-invasive, and you may already have it on your wrist. And you can probably do the same thing with cell phones as well. So okay, there you go. So this is just using accelerometer data over a short time period, maybe over a week. And so I think this could be something that could be very useful, especially if you're trying to explore what is the best thing for my health, because aging is one of the best predictors of whether you're going to have health decline or not. So if you could see like, oh, your biological age as measured by a wearable or some accelerometer in your cell phone is decreasing or increasing depending on what you've been doing, like, oh, maybe you stopped exercise and your biological age goes up. This could be a strong motivator, first of all, to encourage you to have a healthy lifestyle, and also show you what is having the best benefit for you in terms of health as well. So yeah, I completely agree. That's
totally makes sense. Interesting. Yeah. Let me ask another. Think about this. Just kind of brainstorming. I mean, in the future, let's just say we're talking like long term, like 100 years from now, the gene therapy technology is so good, it's phenomenal. Like, what does that look like? Like, what is the end? Like, would it be we've edited every single cell in the body where our genes are now perfect? We have none of our original genes, they're all new and improved, or say 1,000 years or 1,000,000 years. Like, what would be like the perfect gene therapy technology? Or can we even conceptualize that? What are your guys' thoughts on that?
Well, I'm holding great promise in plasmid type gene therapies, as well as the other kinds that organizations like Liz's BioViva and Integrated Health Systems are doing, and some others. But there's so much in the future we just don't know. But with clarity, knowing the end result we're seeking, well, how are we going to get there? We'll figure it out.
It's a very good point.
Like I mentioned, I'm going for a star shot, open-ended life, healthy, of course, and contributing to the world.
Yeah, I mean, I think, and feel free to share your screen. I think in the long run, and I'm just, obviously there's so many unknowns, there's so many paradigm shifts that will happen between now and then. But I assume that eventually we're going to be able to simulate biology very efficiently in the machines, and we'll be able to determine what is the optimal genome, personalized genome for each individual, like you alluded to before, Johnny, like personalized gene therapy for that person to alter their genes in a way that is very robust to make sure that their body is ultra efficient from its ability to operate, from each level, cellular, tissue level, organ system level, and then full body. But go ahead, Gus, share your thought there.
Well, it takes us back to ethical considerations and the big question, why? What will you do with all that extra time? Find a purpose, be a force for good. Otherwise, what? We're just going to play for years and years to the future? No, we got a world to save out there. And here are some of the things that need, that the world can be improved, that once we saw, I got to focus on aging right now, but once it's solved, well, then there'll be time for other things.
I agree with that. Those are all on the next on the list too. Once we can solve this problem.
Yeah, I have a couple questions. One is on the current cost of human genome sequencing. So Johnny, I know you've been looking at this a lot. So what is the current cost of human genome sequencing? Because it's tough to tell, because it seems like there's a variety of options.
Oh, do you mean through the two sources that I showed you?
Yes, that or anything else you found.
Oh yeah. Well, let me scroll back to that page here.
I can answer that one too if you like.
Yeah.
Yeah, it's a few $100. Nebula Genomics is an example, which is a George Church company. They charge, I think it was on sale. They have 3 levels basically. And really you're going to spend a few $100 depending on the level you want. Like the cheapest one, I think was like 150 plus a little bit, maybe 100, 200 more for like the lifetime subscription. For the mid-level, I think all in, you're going to end up paying about $500 total, like probably about 200 for the sequencing, and then 300 for the, oh yeah, here we go. Yeah, I can lay it out here.
Yeah, well, you have to read though, that's the base price, but you also, I believe, are required to sign up for the membership, which is, seems to me, I haven't seen it yet, but it seems to me like it would be worth it and would give you useful insights that you can use for your own health and well-being.
Absolutely.
So that's the program. I don't know about this one. It's certainly getting up there. But I've heard, oh, and here is the Dante website. I don't know if we can find the costs here. There's probably a buy now button or something similar. Oh, shop. Here we go. This should take us right there. No, thank you. Okay. Yeah, US. Research clinical. Well, it's here somewhere. The pricing is probably about the same.
Let's go back to the genomics one, the Nebula one, real quick.
Yeah, I just want to say it blows my mind because this is $100. I can see it's $100 for whole genome sequencing. So this is something I wouldn't even have expected to be the current price. So that's pretty much unbelievable to me. I don't know whether it actually cost them 100. I mean, maybe they're taking a loss on that, but that's incredible.
Yeah.
So,
I
believe this is one of the reasons for the price difference. There's a concept called deep sequencing, if I understand correctly, that they sequence it multiple times in order to arrive at a more accurate result. And I'm probably a little off in the proper definition, but surely there's a reason this is more expensive than this one. And I'd go with this one.
That's 100% correct. I actually did the deep one myself. Out of these 3, so what Nebula essentially promotes is they say that they're looking at much more of the DNA than something like 23andMe. They have on their website this picture that sort of alludes to that says, hey, we're scanning essentially all of your DNA, and other organizations are not scanning quite as much. And comparing those 3, it's really like you were alluding to, Johnny, like how many times do they actually scan the DNA? The ultra deep one, I believe it was 100 times, and it produces 300 gigabytes of data. While the mid one does 30 times and produces about 100 gigabytes. And for most people, unless you're looking for a very rare mutation, the mid one there for the 250 plus another 275 for the membership, that's going to be good enough for the mass majority of people. However, like I said, if you have a specific variant or specific mutation, you think you might be predisposed to something, or you really want to dig ultra deep, then you're going to want to go with the most expensive one. But that's up to you.
Yeah, I've heard that there is either upcoming soon or maybe right now a $100 sequencing service. Yeah, you get what you pay for, but I would buy it and I would compare it with the Nebula and see how it compares.
Absolutely. I mean, the cost of these tools continues to go down. I mean, think about how much it costs for the first genome, probably cost billions. And one of my friends, Dr. Jose Cordero, he always shows this picture that shows the prices over time as they've gone down over the years. And we're looking at this decade seeing genome sequencing so cheap, probably $10 to get your genome sequenced during this decade, which is just like you were saying, Kyle, unbelievable.
Yeah. One other question I had was, Brian Johnson recently came out with this blueprint rejuvenation, and Johnny, I'm not sure if you've heard of this or not, but basically it's taking the best scientific evidence and then trying to translate this into the real world, working with a lot of physicians and scientists to create the best possible protocol to maximize your health and longevity. And it takes a lot of mental fortitude. There's a lot of really strict diet and exercising that I can't do, and I'm pretty good at doing some of this stuff. But have you looked at that and thought about, like, what is he missing in terms of personalization? So is there any big things from personalization you're seeing that may even add on to what he's doing?
Well, yes, I have looked at it, and I do have it to study it more deeply near the top of my long list of things that I need to do. I am impressed that he at his dedication, and that he utilizes the testing systems that Oliver Zollman, a very talented, kind of amazing UK MD, implemented. It's just such, multi-organ system. And so that's what I know about it. But no, I have not tried to find ways that he might improve. But if and when I do, I would certainly make that suggestion to him.
Yeah,
I think that'd be
One
great.
thing I do want to just point out, one concept. This is a bit of a sidebar. The concept of finding your own Achilles heel and fixing it. My great friend and among the best scientists, the best scientist in creating self-directed age management programs and really understanding the science and biomarkers and objective measures, was in great health, but he died abruptly because of a condition in the gut called Merkel's diviculotum, which is, I kind of think of it as a ruptured appendix. It was probably a genetic anomaly in the gut that over time, it burst, and 6 hours later he was dead. So no doubt that, as we grow older, our gut thickness and, organ size, bone mass, on decreases. So that may have been a problem. But my point is that something I like about Oliver's system is that he would find an individual's weak spot, Achilles heel, so to speak, and that person would give special attention to it. So I'm off that sidebar now.
yes, I'll say a quick comment as well, and I definitely agree with your comments there, Johnny. And I think when you look at Project Blueprint and what Brian Johnson has put together, I think that one of the key parts is he's combined measurement with protocol. And you measure something, you then develop a protocol based on those measurements. And I feel like that's the key part. You're developing these interventions based on measurements. So I think in general it is intended to be personalized, but I feel like also that over time will become more personalized for him. And he kind of points out too, I mean, he's came up with something that works, and he says, hey, this is not necessarily the best. This is something that works for me. There's going to be other versions, and we want to iterate and improve upon that. And I'm sure, like, using something like genome sequencing, which I'm confident that they're going to be looking into, including gene therapy as well, with developing a clinical guideline and being able to integrate these new tools into his protocol. I think that they're definitely doing that, and I think that you do need something personalized to really work, because everyone's individual. What's good for me is going to be bad for someone else. And to Johnny's point, like, they did find one of his Achilles heels. Apparently there was something in his vein through his neck, and suddenly they said, okay, we got to focus in and figure out what's happening here. He said, in one of his YouTube videos, said he never knew about this before these doctors were doing all these measurements. So I think as you go through the measurements, that's where you're going to make these discoveries, and that's where it becomes more and more personalized. And I think with what he's built, and I know I've probably talked to you a little bit about this, Johnny, we really want to expand. I think his approach is the way to go because it's a multi-pronged rejuvenation protocol. And we really want to expand on that. And I think a lot of companies they should take that protocol, and which is all public. He laid it out all for free too. I think it was tremendous. I think tremendously valuable. It's all right there on the website, on his website, Project Blueprint. And I think we can iterate on that and incorporate new tools and techniques into that protocol and basically make our own personalized one. And I feel like that is the path that will effectively allow us to reverse the aging process, or at least dramatically slow it down, depending on, oh yeah, thank you for bringing that up, depending on what timeline. So he went back 5.1 years on his epigenetic age. Across some biomarkers, he did go back decades. I actually believe that you can go back decades across every single biomarker eventually. It just depends on how advanced our technology becomes. And we're actually working on our own protocol now using a physics-based approach, and we want to use a similar methodology, actually, where you're measuring the biological age across all 78 organs and the microbiome, like you were talking about your friend with his gut. There is this gut-brain axis that there's this communication between the gut and the brain, and it's an area of research. The microbiome is actually a wide area of research that still a lot of things that are needed to be discovered, but a lot of companies, they're specifically targeting the microbiome. And I think, we can take measurements of young athletes, 21 to 24, and we can look at where do they lie? What does their microbiome look like? What are their blood biomarkers look like? And then we can actually apply, say, a protocol similar to Brian Johnson's, but some variation. And then we could see, if it's someone who's a baby boomer, say 65 and above, we can have a direct mapping. We can say this person is this close to what we think of when we measure someone who's highly athletic and in the best shape of their lives to some degree. That was really great, Johnny. I just have a quick comment and question.
Yeah, please.
It looks like from what you were just showing, that was really interesting. It looks like there's what you're kind of building there is a map of all these genes to the specific phenotypic expressions, like you were saying, like sprinting or cognitive ability. So it sounds like we sort of have some targets. Is that the idea? We have some targets that maybe in the future we can use gene therapy to alter?
Well, yes, absolutely.
Nice.
And if you liked what I just showed you, I think you're gonna like the Okvar report.
Awesome.
Any more questions?
Jason,
Kyle?
no, I think I'm good. Kyle, anything?
Okay, good.
Yeah,
So
well, I have something that's relevant. So this week was the third International Summit on human genome editing, and basically one of the major conclusions there was we're not ready for germline editing. On the other hand, there's a big motivation from parents that's always there to secure the best future for their children. And in many cases, doing germline editing would be quite extreme. But in many cases, there's a high chance that someone may pass on a rare gene that could be detrimental for children. And so I think this is kind of coming into conflict. Have you thought much about this conflict and how it may be resolved? Many of the things you're talking about may give slight advantages. And if it was considered normal to live 100 and someone lived 80, and you could maybe make a gene that could make them live to on average 100, maybe that would be considered something they can do. But there are things that are considered more detrimental and things that are a little bit more minimal. So do you have any ideas of how we get past this? Because it's both a scientific and ethical question. And I have no doubt in the future that this will be overcome but I'm not quite sure of the path. So have you thought about that issue?
Well, yes, of a great many things that I think about while needing to stay focused on the most important things. That is, in fact, one of the things I have given a good amount of thought, and I have collected a number of objections to aging interventions and solutions, objections to gene therapy. But to answer your first question, I'm convinced that that there are solutions to them, and just like the multiple of tens, dozens, hundreds, thousands of similar questions that have plagued mankind since ancient times, that we can solve them. So that's my answer to your first question. And please refresh me on the second question if I did not yet answer it.
No, that's pretty much the question. And I think the main issue that I kind of see happening is, this was highlighted at the summit this week as well, is that basically someone already attempted gene editing. It's unclear whether it was successful or not in China for these twins that may be susceptible to HIV. The question of whether they even were to begin with is suspect. But in any case, I think that the fact that this was done without the knowledge of anyone, then it was kind of announced publicly, it created a huge problem because we never really knew if this was safe to begin with. And then on top of that, it was a question of whether it was even needed. So my thought is, like, how do we do this in the best way possible, where we make sure that all the objections are heard and really assess that this could go forward safely? Because one, like I was talking about last week, in an early gene therapy trial, there were several problems, and this was within a regulated clinical trial that everyone knew about, and that led to a lot of problems in the gene therapy field. And I kind of hope that this does not reoccur. But we had this case in China where kind of a very similar situation reoccurred, and luckily, it seems like nothing really went wrong safety-wise, but it potentially could. And so this could really be detrimental to the field. So I'm just trying to think of ways to make sure that we do get past this in a way that everyone is okay with, and a way that's safe. So things are going to go wrong in clinical trials no matter what, because we never can understand what's going to be the outcome of biological interventions, whether it's gene therapy or otherwise. But I just want to make sure that we're doing this in the best way possible to ensure the safety of others.
Yeah, I'll say a quick comment. 100% agree. And with any new technology, whether it's gene therapy or AI or anything, it's to some degree and always a double-edged sword, where we're building increasingly powerful tools that can do a lot of good, but they could also do a lot of bad, especially as the accessibility and cost comes down. If someone has, these tools in their garage, they could create a dangerous virus that could be highly communicable and more deadly than the coronavirus, for example. There could be other, dangerous things that could definitely come about. And to your other point too, there are a lot of ethical considerations, and a lot of people might say, should we alter, if someone's going to have a baby, should we alter their germline? Should we be able to do that? And I think I think what society will actually end up saying is that it's okay to do, because you're basically saving a lot of different pain and suffering of that future child's life, especially if it's, like you're alluding to, something very detrimental. But there's always going to be a trade-off, and I think there does need to be getting a lot of people and organizations on board. What are the regulations? What are the ethical considerations? And I think the main thing is like having that open communication across governments, across corporations, and across people to really get everyone on board, with how we're going to go about doing this, because otherwise, there could be some chaos for sure. So,
well, we certainly want to proceed with the highest ethics. There is one fact that, to my mind, is settled. Aging is 100% fatal. And in our life, in our own health programs, we must make a calculated risk.
Well, I have to go,
Well,
guys. If you want to continue talking, but
Thanks, Kyle.
very interesting, a lot of very interesting information, and I'm going to be thinking about a lot about it. So, thank you for the overall overview.
Thank you, Johnny, and thank you, Kyle. Really appreciate it. And yeah, thanks again, guys. Really phenomenal.
Bye, guys.